Hello everyone. Today I'm planning to talk about the controversy of the topic water fluoridation. I guess if you are watching it, this means you already heard about it and know something. So I will not go into detailed introduction. I'll just read you one sentence from Wikipedia and then we go straight to main question. So water fluoridation is a controlled addition of fluoride to public water supplies to reduce tooth decay. That was the intention. And it started in US in 1945. But notice here the dose. The recommended fluoride levels is 0.5 to 1.5 milligrams per liter. In US, they set the standard to be 0.7 milligram per liter since 2015, which got lowered from 1.2 milligrams per liter. You see, it got cut almost in half. Here is a confirmation. The US government sets optimal fluoridation at 0.7 milligram per liter and safety standard at 2.0. Okay, so this is numbers which we will need to have in mind because everything depends on the dose crucially. 0.7 optimum level, 1.2 previous optimal level, and 2.0 as a safety standard. So 0.7 is what they're adding on purpose. Now I get to the main part, I would say. So we need to evaluate the effectiveness of this procedure and the safety of this procedure. The effectiveness originally was pretty much clear, but recently, you see, there was a Cochrane review on this topic, and here's what they found. The team of researchers from universities of Manchester, Dundee, Aberdeen reviewed the evidence from 157 studies which compared communities that had fluoride added to their water supplies with communities that had no additional fluoride in their water. They found that the benefit of fluoridation has declined since 1970s, when fluoride toothpaste became more widely available. Yeah, that's kind of expected, because why would you need to consume fluoride inside your body, into your body, if you could just use it in a toothpaste? So the practice is kind of obsolete anyway. However, US still hands to it, and CDC and World Health Organization continuing to recommend it, and they're very proud of it. They also, by the way, studied the side effect of this water fluoridation practice, which was also known for many years. It's called dental fluorosis. In case you have too much of fluoride in water, it could happen. But in my opinion, it's kind of a straw man saying that, oh, I hear it all the time, that even on Wikipedia, you see something like too much fluoride could be bad because of dental fluorosis. However, the biggest concern, which I know people have, is actually neurotoxicity of fluoride. The possibility of brain cells damage and lower IQ. And that's what I'm mostly planning to discuss today. So I found several reviews of bunch of studies on this topic. For example, Here is this review by Grandjean. What he is talking about here. Systemic fluoride uptake is suspected of causing adverse effects, in particular neurotoxicity during early development. The latest supported by experimental neurotoxicity findings and toxicokinetic evidence of fluoride passing into the brain. And here is the result. Neurotoxicity appeared to be dose-dependent, and tentative benchmark dose calculations suggest that safe exposures are likely to be below currently accepted or recommended fluoride concentration in drinking water. Well, if I look at the studies which he used, just how they are, these two tables, overall, most of them have concentrations higher than what CDC recommends as the optimal level. And a couple of studies which have the concentration lower didn't show any significant result. But there's very few of them. So you could scroll through the whole table. You see that we are comparing all these numbers somewhere above 2 milligrams per liter. Yeah, they either use water or urine. That's approximately the same. So I tried to find another review. Here it is, for example. They kind of do the same. In something they're slightly better because they also add the risk of possible biases. But the problem kind of remains the same. Most of the studies are testing something, from 2 and above, sometimes from around 0.701 and above. So yes, some of them showed some problems for the previous optimal level, which was, I guess, in effect for 60 years, and that's probably the reason why CDC dropped it by half. Yeah, for 0.7, there are almost no evidence here. There is a third review which I found. Here is a PDF version of it. I don't know whether you could see it, or is it too little? Let me zoom in a little bit. Yeah, I like it even more because instead of listing biases as low high, they literally tell you how the study managed the confounders, what exactly they control for, so you could decide for yourself whether the study is good or bad. And again, a lot of data here shows that fluoride lowers IQ if it's above 2 milligrams per liter, and in some cases above 1.2, right? Unfortunately, there's less studies for something around, comparing like 0.15 versus 0.85, right? That's actually what we would need. In this case, didn't find the effect. The only study where I found they had some effect, I think, was this one in Canada, and they control for many factors. So overall, yeah, there is a result here. The study is a little bit complicated because they're comparing IQ of kids for mothers who are consuming fluoride, and they have two different methods of breastfeeding, like two different methods of feeding kids: breastfeeding and formula feeding. And both of them, they have high fluoride group and small fluoride group. Essentially, the main point is that, you know, this baby formulas contain a lot of fluoride, and there is some fluoride getting through breast milk through mother who was drinking fluoridated water as well, but it's kind of small effect. So we have four groups, you see: breastfed fluoridated, breastfed non-fluoridated, formula fed fluoridated, formula fed non-fluoridated, right? And they were measuring two types of IQ, verbal and performance. I don't know what's verbal, it's something weird. And as you can see, there is some reduction in both groups in the IQ, especially in the formula fed group. 99.7 versus 105.6. However, again, also have the standard deviation in mind. So you need to think about this sort of two Gaussian peaks, which are pretty broad, almost overlapping like this. And for the fluoride concentrations, where are they? Here they are. As you can see, 0.58 versus 0.13 and 0.59 versus 0.13 milligrams per liter. So yeah, both of them are below what's currently recommended as the optimal fluoridation level. So this effect is preserved even for small values of fluoride, small concentration of fluoride. But again, it's kind of only one study, and unfortunately, yeah, unfortunately, most of the studies are done on this useless interval. It's almost like if the whole body of researchers is afraid to test the interval which is of the biggest interest to the public. I guess it's a part of the dysfunctionality of academia, that they create this illusion of consensus, illusion of intensive study, but in reality it's just only one or two studies targeting the interval of interests. And as option, maybe that there's no significant effect for this dosage. Yeah, you see, like another review also targeting the wrong interval. Yeah, I would like to also make an important comment that even if we have a lot of evidence suggesting that the fluoride concentration twice as high as what we consider safe today already leads to problem, it doesn't mean that water fluoridation is safe. That's not how we usually evaluate the safeness of any drug. If your safety concentration is just 2 times smaller than the safe and effective concentration, which you should consider to be safe 2 times smaller than already dangerous one, which leads to problem, usually you only use this drug if you really need it and you can't avoid it, which in this case is totally not true, because you have toothpaste. Why? First of all, you don't really control very well the dosage which person takes. Somebody could drink, 3 times more water than average person. You also don't really know the interaction between this drug and other potential chemicals which person could take as other drugs or environmental factors. So this interaction could make it way more dangerous. And finally, there might be smaller doses, way more subtle changes, which is just more difficult to track with studies. So I would be cautious about and drinking fluoridated water based on this kind of reviews. Yeah, so that's only correlation studies. That doesn't tell us about anything about how it works, right? So this means it doesn't give us causal connections. I would like to talk a little bit more about that as well. But I would like to make a certain important disclaimer here that basically biology is not real science. Whenever biologists do something in vitro and we found some effect, it has nothing to do with what actually happening in the body, and whether the effect they found is actually responsible for the pathology we see. Okay, so it's all hypothesis. Luckily, we also have animal studies, and that's way better because animal is already the full organism, right? So this whole immune system is everything working, which is kind of close to human in some sense. And yeah, we could at the same time cut its body, euthanize it, cut its brain, find everything, see everything explicitly, and on top of that we have perfect control. We could have intervention group, control group in exactly the same environments. So we kill all the confounding factors immediately. Yeah, so let me talk about them a little bit. They're also mentioned in the review here. Let me find experimental neurotoxicity. So let me first read the general part about what this review kind of found by looking at the literature. In vitro studies have documented fluoride toxicity to brain cells. Most of the studies using high fluoride concentrations, so some effects have been demonstrated at lower, more realistic levels. In the low dose studies, 10 micrograms per liter was sufficient to induce lipid peroxidation and result in biochemical changes in brain cells, while 57 micrograms per liter induced inflammatory reactions in brain cells. These concentrations are similar to the upper ranges of serum fluoride levels reported in general population. So in case you didn't know anything about this concentration, you'd be like, what is these numbers? Right? They just trying to make the case that this is the numbers precisely relevant for us. In addition, yeah, fluoride can negatively affect brain development threats at levels below those that cause dental lesions. Okay. Yeah. Okay. Utilizing computerized surveillance of rat behavior, a landmark study showed signs of neurotoxicity at elevated fluoride exposure. And more recent studies have reported fluoride-induced neurochemical, biochemical, anatomic changes in the brains of treated animals. Also often those much above human exposure levels. Yeah, they're talking about potential toxicity in the thyroid gland and endocrine disruption. Yeah, here's they're talking about National Toxicology Program, where they focus on fluoride and toxicity in regards to learning and memory. At water concentration higher than 0.7 milligram, found a low to moderate level of evidence. Okay. Here, there's a very important comment to make. Most experimental studies had used concentration exceeding the levels added to water fluoridation programs, but the NTP recognized that rats require about 5 times more fluoride in their water to achieve the same serum fluoride concentration as humans. It's very important because I already see in person making a review of the study without realizing it. Okay, look, for example, I'll show you what he says.
In a different study, this time focusing on animals, it was tested whether fluoridated water affected the cognition of rats. One group of rats was given fluoridated water and the other had non-fluoridated water.
Many months later,
The rats who drank water with fluorine had a noticeable drop in learning and memory capabilities. Surely, this is proof that fluoride affects IQ. One small problem. The rats were given water with fluoride concentrations 5 times higher than humans consume. It's a miracle that only cognition was affected and the rats didn't flat out die. Failed. Okay, but what?
Why? That's the whole point. They did it on purpose to match the concentration in the blood, which is what's relevant. I guess you just haven't read it carefully. Okay, where I was. The review. Okay. Yeah, subsequently several additional development studies have been published, including 2 that reported impaired learning memory in rats consuming water with fairly low fluoride concentrations. However, not all studies have reported adverse effects. Okay. That's this part I already overall reviewed. Yeah. So, but he didn't talk that much about what's happening there. So I found another review where they try to figure this out. You see impacts of fluoride neurotoxicity and mitochondrial dysfunction on cognition and mental health. Literature review. Okay, so here's the abstract. Okay, clear what it did. These studies observed physical changes, fur loss, and delayed reflex development of animals, intelligence loss, increased hyperactivity, and irregular moods associated with fluoride exposure. Okay, very well. Here they're explaining what the sources of fluoride. Okay, results. Fluoride and neurodevelopment, animal studies, cognitive function. Yeah, so I skipped this part, but they explained that there are several substances we add, and we could add to our water supply, either this or this or this or what. Yeah, so there was some three. They should be 3. Sodium fluoride exposure in animals could be linked to cognitive, behavioral, and memory disruption. Okay, so more severe neurotoxic effect been observed among still developing rats. Okay, statistically significant impact on mouse cognition and behavior such as slow spatial learning, depressive tendencies, and anxious tendencies. Short-term and long-term memory impairment has also been noted. Yeah, prenatal fluoride exposure in rats, so it means parents, right, was associated with hyperactivity compared to that induced by amphetamines. Yeah, okay. They think it's because the blood-brain barrier of young rats is not very well developed yet. Okay, sex differences. Well, yeah, they're talking about the same kind of problems: hyperactivity, ADHD, learning ability. And by the way, with quite low levels, again 5-10 milligrams per liter. Remember, for rats, you need to divide numbers by 5 to get the human levels. human equivalent. Then behavior, they're talking about serotonin increase. Yeah. Yeah, serotonin is a neurotransmitter that, when present in the brain and at inadequate levels, is heavily implicated in the development of depression and anxiety. Yeah. Okay. Increased excitement in hippocampus, impaired memory, anxiety-like behavior. Also, again, that might be due to passage of fluoride through blood-brain barrier, causing maladaptive changes to the hippocampus. A human study, that's we already reviewed, that's IQ impact on kids. Again, not very conclusive, and we need more studies at the right dose. Mental health, and here we finally get into the ideas how it works. Several studies have linked fluoride exposure to mitochondrial dysfunction. Okay, mitochondria energy producing membrane bound organelles that produce most biochemical reactions within eukaryotic cells. So I guess you know what mitochondria is. Mitochondria serve a variety of purposes, including regulating metabolism and apoptosis. Apoptosis is cell death. And then they contain mitochondrial DNA. Okay, you know it. Okay, cancer. Okay. Yeah. While many studies of both animals and human responses to fluoride exposure have found evidence of neurotoxicity, a mechanism of this damage is not usually agreed upon. Some studies attributed an association between neurotransmitter levels, fluoride consumption, to clinical neurotoxicity. Others emphasize changes in neural anatomy, and others suggest mitochondrial dysfunction as a potential mechanism. You see, okay, as I was telling you, there is no agreement. They don't know anything. It's all mess. Okay, so structural changes. I don't know what this is. Low levels of mitochondrial DNA. Yeah, okay. Yeah, that's already they explained it. In mice, sodium fluoride concentration of about 5 milligram per liter, which again for humans would be around recommended dose, have been shown to lead to oxidative stress and inhibition of antioxidant enzymes. This leads to high concentration of reactive oxygen species, causing mitochondrial damage, including lipid peroxidation, mitochondrial membrane depolarization, cell apoptosis, okay, which means death. So I guess we're burning too much oxygen, creating this reactive oxygen species. That's not good. That's like burning out our cell. Okay, see, ROS can also result in degradation of mitochondrial DNA. Yeah, furthermore, sodium fluoride is suspected to lead to autophagy deficiency, apoptosis augmentation. I already don't know what it is. Compromised neural survival, membrane loss, increased permeability, and reduced oxidative phosphorylation in the mitochondria of rats. Okay, I don't know what this is. Yeah, separately, various causes of neuroinflammation are also of interest to researchers. Neuroinflammation may occur because of fluoride intake, resulting in potential neurological and neurodegenerative disorders based on dose of fluoride received. Okay, they're talking about what kind of chemicals, what kind of enzymes are more expressed in rats which receive higher dose of fluoride. Yeah, for example, vascular and arterial growth factors. It's a thing which is created when you grow your endothelial or inside your veins, right, and arteries, which kind of indicated you're kind of damaged, right, and they think it's might be a marker of inflammation, in this case neuroinflammation, because probably it's all in the brain. Okay. Finally, they talk about back on neurotransmitter levels. We already read this. Again, serotonin levels have been shown to significantly increase in the brains of rats following fluoride exposure. Yeah, we already know that. I don't know what's neuro to A cells in mice, similar concept of observed cells that have been exposed to fluoride for 24 hours were observed to have swelling, agglutination, vocal formation, edema, and loss of synapses compared to controls. The release of neurotransmitter glutamate was disrupted and structural damage occurred within the cell, supporting the idea of fluoride-induced neurotoxicity. Another pathway that is under investigation for fluoride-induced effects is this thing which is responsible for creation of new mitochondria, I guess. Okay, I guess at least continues. I don't want to read more, but again the same. Reactive oxidation species, mitochondrial damage, apoptosis. Guess you get the idea. The same. Talking about mitochondria. So if you want, there's more details on each of these questions. I opened some of the studies which they discuss. Here's a PDF. Prolonged exposure to high fluoride levels during adolescence. And the functional impairment in the hippocampus. That study done on rat, 21-year-old mice received 10 or 50 milligram per liter. Again, need to divide approximately by 5 to get human equivalent, which means around the safety standard dose for this is around safety standard dose for OCDC. And what they found, mostly memory tests getting worse, where other see some sort of Morris water maze performance. It's some sort of cognitive test they use. I guess there is some effect there, and guess another test that you don't want to get how it works. But I guess more interestingly, you could look at the size of the brain. You see the effects of fluoride exposure from adolescence to adulthood on the mature neuronal density in most hippocampus. And here you look at different parts of hippocampus. The one which we see some decrease, as opposed to increase, by the way, which is strange, is this dentate gyrus. You see this part. Even for this small dose, there is some little effect. I look at that what it is. It's a region within hippocampal formation of the brain, playing a crucial role in memory encoding, learning, and emotional behavior. Okay. I lost it again. Here's another study. It's about serotonin. Like they told you. I guess I will not go into much details. I think you probably could read for yourself. I know, do they have like results? Results. Mice exposed to 50 milligram, right? Yeah, but that's too big dose, even if we divide by 5. Activity. increased hair loss. So it actually helps hair loss. Okay, so I just showed it because it was mentioned. Yeah, both peripheral and central serotonin was increased in the fluoride treated mice. Here, that study which I wasn't able to get access to, but more or less it's clear what they're doing from just an abstract. Yes, they exposed rats to, as you can see, 10 ppm parts per million, which is again 1 part per million, approximately 1 milligram per liter, and 50 ppm of fluoride. So this approximately would corresponds to 2 milligrams per liter, looking at this for something huge. Again, they run through some sort of usual tests. Then after 60 days they euthanized them, analyzed some biomarkers, and what they found. They found impaired locomotion, memory impairment, and behavior resembling depression in the animals were evinced by reduced mobility index. Okay. Additionally, alterations in antioxidant levels and oxidative stress parameters were observed in the brain. The expression levels of various apoptotic and inflammatory biomarkers showed apoptosis in neurons. Okay, confocal studies showed increased expression of inflammatory, apoptotic, and mitochondrial dysfunction markers for fluoride-treated animals. Yeah, toxicity was more prominent in 50 ppm fluoride-treated animals. Well, yeah, of course. I wonder what the results for 10 ppm are. That would be more relevant because of the safety standard of CDC. But I can't open the study. Okay, here, yeah, that's a good one because they use really small dose. You see, fluoride concentrations from 5 to 10 milligrams per liter. Again, also for rats. And this 5 milligram would correspond to 1 milligram, right, for human. So this means it's exactly what we want with our optimal levels, right? Remember what CDC adds on purpose in water. So that's very interesting. Overall, again, they found what the problems with some sort of memory, learning. You see, step down inhibitory avoidance tests, latency to step down. Well, you could read about how exactly they're testing, the smartness of the mice. It's really funny, but you see, even for 5 milligrams, performance goes down. For 10 milligrams, it's even worse. Same here, immobility. You see, immobility goes up even for 5 milligrams. Swimming goes down. Climbing doesn't change. Very interesting. Okay. This I already forgot what it is. Here, oxidative stress markers. That's we already discussed, yeah. But it became very weirdly depending on the part of the brain. Again, what they in hippocampus they go down with concentration of fluoride going up. Yeah, not sure how to interpret it. Is it good? Is it bad? And this is transaminase activities. which again, in some parts of the brain, is strongly affected even by 5 milligram per liter dose. In case you wonder what this is, I also look it up. Transaminases are a group of enzymes that play a crucial role in amino acid metabolism by facilitating the transfer of amino groups between amino acids and keto acids. This process, called transamination, is essential for both the synthesis and breakdown of amino acids, which is the building blocks of proteins, right? Okay, so basically it's the most standard protein metabolism. You need to constantly form new proteins. This means you need to form amino acids to build them, and you need to degrade them. Yeah, and this is heavily suppressed in some parts of rat's brain. I don't know, it looks kind of a little bit scary. This is other some enzymes which I already don't know, but yes, you get the idea. You could read this paper for yourself. I like it because it has the relevant dosage, finally. Lastly, there is some study about nerve conductivity, nerve conduction velocity, again also in rats. Here, I also don't have access to it, but the concentrations were like quite high too, 10 milligrams per liter, 100 and 150. And of course, in this group, they reduced the lower nerve impulse conduction velocity, but they say that in general there was a negative correlation between the concentration of fluorides in plasma and the nerve conduction velocity. Yeah, and they say that potential may explain the deficits in learning and memory tests that have been documented in numerous studies evaluating individuals exposed to fluoride consumptions. Yes, so we already looked at them all. So I guess that's more or less it. I don't know. I haven't found that much more. As you can see, the results are a little bit, non-decisive. For doses slightly bigger than recommended, we see some problems already in humans. In rats, we see problems for the same dosage as well, and even for the optimal ones, more or less for the same dosage in the blood. And I guess the additional studies I needed to say for sure. But this is a bit strange, because if you look at this video, which I found, how this all happened, which a little bit gives a little bit negative connotation on this topic, but you could welcome to find something better. There was no proper testing whenever they were implementing this practice. All this research come later, post factum. There was no testing, there was no voting. He also mentioned there is some corruption involved. I don't know whether it was essential. I guess what was essential is this kind of idea that we want, public to be safe, public to be good. We want the government to solve problems and so on. And if you read like any review, pretty much, it praises this practice. That CDC, World Health Organization, they all praise it. For them, it's like it's one of the greatest achievements of health sciences, or like healthcare system. And again, they were pretty toxic about it. They were smearing people who speak out against it. So I'm afraid now, when Cochrane review already found that it's kind of useless, they could not back up. You see, right now I'm showing you review which criticized the Cochrane review, trying to find gaps in the logic. I mean, they're pretty not convincing, but it's more like they could not admit that this is wrong, that this practice is useless and most likely harmful, because again they were not behaving decently, they were smearing the same views, and now they have to double down, because it never was about fair science for them, it was about ideology, and the ideology, as you can see here, is pure collectivism, right? Further, with public health intervention, the issue is not about individual patient benefit, but whether the population as a whole will benefit. Yeah, this phrase is just wrong. It doesn't make any sense. There is no populational benefit different from the collection of benefits of each individual that the population is made of. This is pure collectivism and pure evil, and that's what these people are. I hope you enjoyed this video. See you next time.